Thank you very much. So I'll talk for about half an hour, and then we've got time for questions and discussion. Thank you for inviting me, and I'm sorry that we're not doing this in person, but we'll do our best. So let's just, before I begin, this is by way of acknowledgement, really, that the research you're going to hear about is the result of collaborations, and lots of names on this slide. But the first group are people involved in psychology and brain scanning, MRI studies. The second group are particularly looking at hormones, and the theory that we'll be looking at are prenatal sex steroid hormones, exposure to different levels and how they change brain development and likelihood of autism. But that group also includes people working at the genetics level. And at the bottom of this slide are collaborators in Denmark who have enabled a lot of their prenatal sex steroid work. So briefly, just to make sure we're all on the same page, what is autism? So in this image on the top right, you can see a child playing alone, and the diagnosis is made on the basis of social difficulties and communication difficulties. The fact that it's a young boy who's playing may become relevant because autism is diagnosed in boys much more often than girls. But you can see that apart from the social communication difficulties, the other diagnostic criteria are difficulties adjusting to change, especially unexpected change, and then the development of these very narrow interests, sometimes called obsessions, and unusually repetitive behavior. And in the latest diagnostic criteria, they also include sensory hypersensitivity. So here's a little bit about prevalence, and here's where you can see the link with your gender. But you can see that autism, this is 2014 data from the Center for Disease Control in the US. So it's not the latest prevalence data, but it's pretty similar to the latest data, showing that autism is about 1 in 48 males, 1 in 189 females. So that's a sex ratio of 4:1, male to female. And if you combine both sexes, it's about 1 in 68 kids. The graph shows you that autism can be diagnosed across the IQ spectrum. So, some autistic people just have autism and others have below average IQ, so they have additional learning difficulties. What I've put on the left of the slide is that the likely sex ratio may be more like 3:1, because probably a proportion of females are not getting diagnosed, they're being missed. And increasingly more females are coming forward for a diagnosis. So the kind of prevalent studies may be somewhat underestimating the number of females that need a diagnosis. And what you can see here is that if you look at the age at which males and females get their diagnosis of autism, females are being diagnosed later than males, suggesting that maybe the presentation of autism in females is a bit more subtle, or that potentially females are making more effort to hide their autism. This is sometimes referred to as camouflaging, maybe because there's greater social pressure on females in many societies to be sociable, to be communicative, and some stigma attached if you're not. So in terms of what we know about the causes, this slide sort of shows the proportion of variants in autism that can be attributed to either rare genetic variants, which are those small slices in the pie, or common genetic variants, which is that big red section in the pie chart, or variation in autism that is unaccounted by either rare or common genetic variants. And you can see that rare genetic variants, that's to say mutations whether de novo or inherited, only account for a small proportion of cases. Common genetic variants seems to account for a much larger chunk, but you need large samples to demonstrate through genome-wide association studies association with common genetic variants that we all carry. And then, of course, you can see that not all of autism is genetic. So there's kind of non-genetic factors in the light blue part of this pie chart. But in terms of the genes that have been identified in association with autism, there's a very good website. You can see it at the bottom of this slide, called safari.org. It's a charity that lists every new genetic association with autism. And there are at least 100 so-called high confidence genes that have been identified. You can see some of them on the table on the right. These are genes that are expressed in the brain, unsurprisingly. They're called high confidence because they've been independently replicated by different labs. But as I say, many of these are rare genetic variants, which may only be seen in up to 10% of autistic people. And so the other genes that are relevant to autism are likely to fall into genes of small effect, single nucleotide polymorphisms, which in combination might push somebody towards a diagnosis. So I said that the autism genes that have been found are expressed in the brain. And what this shows is that they're expressed prenatally in the brain. So although a child might get their diagnosis in early childhood, and sometimes even later, the causes, the genetic causes that are changing brain development are likely to be the result of genes that are expressed prenatally. So it's changes in the prenatal brain that are relevant to autism. And as we've heard, it's not just, autism isn't just genetic. So here we've got some identical twins, one of whom is autistic and one isn't. So if autism was 100% genetic, then if one of them got a diagnosis, the other one should too. The fact that you can find these discordant sets of identical twins suggests that there'll be some non-genetic or epigenetic factors that are acting on the genes, perhaps changing DNA methylation. And again, this is what's been found just looking at these unusual pairs of twins, differences in gene expression levels between identical twins. So we conducted a study some years ago now, 2013, looking at typical sex differences in the brain using MRI to look at what's called VBM, volumetric based morphometry. So which regions in the brain differ between typical males and females. And then we also did an analysis of which brain regions differ between autism and controls, and then did an overlap. And as you can see, many of the differences between autism and controls overlap with regions that differ between typical males and females. And this is particularly true in females with autism, suggesting that whatever is causing autism in terms of brain development, here we're just looking at brain structure, might be driven by regions of the brain that are sexually dimorphic. This data shows that there are sex differences in brain development in the general population. You can see that on the left with males in blue and females in red, just showing brain volume across age, by age, that males have a larger brain than females in development, larger brain volume. And on the right, you can see that when it comes to autism, shown in red, versus non-autistic kids shown in blue, tracked across development, this time from 12 months old to 72 months, the autistic brain seems to be larger at each point in development. So this has led to something called the early brain overgrowth theory of autism, that the brain in autism might be growing faster than a typical brain. But it also, because of the sex differences that are seen in the general population, it leads to this idea that maybe the autistic brain is an extreme of the typical male brain in terms of its pattern of growth. And when we sort of drill down to using neuropathology, so post-mortem studies to look at what might be causing this brain overgrowth in autism, what you find is more neurons in the autistic brain, particularly in the frontal lobe. This is data from the University of California at San Diego, Eric Corshen's lab, suggesting 65% more neurons in the frontal lobe in autistic brains. And we know from very old studies, again, relating this back to typical sex differences, that the male brain, leaving aside autism, the typical male brain contains more neurons than the typical female brain. So again, these are post-mortem studies across development. So where you have the opportunity to count individual neurons, you can see that the male brain on average has 22 billion, and the female brain on average has 19 billion. So there are differences, it's a 16% difference in terms of the sexes. The other thing we can see in the autistic brain, again from post-mortem studies, is more dendritic spines. So now we're looking at individual neurons on the right from an autistic brain at post-mortem, on the left from a typical brain. And you can see with the naked eye that on the right, the autistic neuron has more of those white dots along the neuron, each of which is the location of a dendritic spine where it forms a connection with its neighboring neuron. So this is suggesting that in autism, there are not only more neurons, but more connections between neurons. And you can see that the graph in the center suggests that this might be due to reduced apoptosis or pruning of neural connections, because if you divide these post-mortem brain samples into younger and older, the dotted line is the autism group, and there's kind of less of a reduction across with age compared to the typical group, suggesting that they're not pruning the same amount of connections as in a typical group. Still just on the topic of how does autism relate to typical sex differences, this is a test of empathy at the psychological level, where you look at the photograph of the eyes and you have to say which word surrounding the eyes, the photograph, best describes what the person in the photo is thinking or feeling. So it's quite a tough test, but the correct answer for this item is that is dispirited, that she's a bit sad. So you're making judgments about what someone else might feel on the basis of minimal information to do with emotional expression in the face. And what you can see on the bottom left is the scores on this test, that women in the population score higher than men on average, and males and females with autism score lower than typical individuals. But again, performance could be, by the autism group, could be described as an extreme of the typical male profile. And on the bottom right, you can see that when people take this test whilst having a functional MRI, there's a particular part of the frontal lobe, the left inferior frontal gyrus, which is more active in women than men when they're doing this test, and which is underactive in autistic people when they do this test. So again, in terms of not just performance on the psychological test, but also brain activity during the test, autism could be characterized as an extreme of the typical male profile. The other psychological test that's of relevance to autism is attention to detail. So this is a this is called the embedded figures test, where you have to find this cube as quickly as you can in the overall display. I'll let you see if you can see it. But a lot of studies over the last 50 years actually have shown a typical sex difference, that males are faster on this test than females. So this is showing the average number of seconds it takes for males to find the target or for females to find the target. And autistic people are super quick and super accurate on this test of attention to detail. But again, more relevant is that autism looks like an extreme of the typical male profile on this non-social test of cognition. And the brain scanning data shows which part of the brain is involved in doing this task. Let's just see if we can see that. It's the parietal cortex, so visual cortex, which is less active in typical males than typical females, and even and underactive in autism despite superior performance. Another way to think about autism is not in terms of categorical diagnosis, but in terms of autistic traits. So we all have some autistic traits. This is a big online study that we conducted of 600,000 typical individuals. And what's shown is males in purple in the general population score higher than females in terms of autistic traits. So we all have some autistic traits. Males seem to have more than females. And in green and red are autistic people who are simply shifted over to the right of this distribution. But again, we could think of their profile as being an extreme of the typical male profile. So let's get to the kind of theory that we're talking about, which is prenatal sex steroids. So these are hormones that lie in this particular pathway. It's called the delta-4 pathway, that starts with progesterone and goes through various steps, each of these androgens being converted into the next one along the pathway, catalyzed by different enzymes. And the idea goes all the way back to a neurologist called Norman Geschwind, who was working in the 1980s in Boston, who argued that these hormones, these sex steroid hormones, have organizational effects on brain development. So the amount of the hormone that you're exposed to permanently changes brain structure and function and masculinizes the brain. And a lot of the work came from animal research, and we'll come back to that. So we've been interested to study this in relation to the development of autism. And the way we do this is to collect amniotic fluid during pregnancy, because this might be the relevant critical period for these hormonal effects on brain development. So we study women who are having amniocentesis during pregnancy, which, as you know, is a clinical procedure that some women opt in to do. It's less common these days, but it was usually offered for women who were suspected as that their baby might be at risk of Down syndrome. And we asked these women for their consent to also analyze the fluid for these hormones. And then we wait for the baby to grow up, and we look at effectively correlations between prenatal sex steroid hormones and behavior, but also, as you can see in this slide, brain structure. So when these kids were 8 years old, we invited them to have an MRI, and this is just showing you gray matter in the brain and showing you which regions of the brain show a correlation with prenatal testosterone. And there are a number of different brain regions, such as the planum temporale, which is a language area, or the right temporoparietal junction, or superior temporal sulcus, which is a social brain area involved in imagining what other people might be thinking. These are really just kind of proof of principle that the hormone exposure is changing brain structure, which we knew from animal research, but hadn't been demonstrated before in humans. But more relevant to autism was that we could look to see whether there was any correlation between the levels of prenatal testosterone and the number of autistic traits the child showed at 4 years old. So the way this study is done is that each of these dots is a boy or a girl. Boys in are little blue triangles, girls, the red circles. They're running around Cambridge. They're not autistic. They're just in the study because their mother had an amniocentesis, which is the only ethical way we can get close to the fetus to measure the hormones. But then at 4 years old, the mother fills in a questionnaire with items like this, which allows us to count how many autistic traits the child has. Remember, we said earlier that we all have some. And what you can see is a positive correlation that the higher the child's prenatal testosterone, the more autistic traits they're showing 4 years later. No surprise that the red dots are all bunched up to the left here because females they produce less testosterone than males, where the testosterone is coming from the testes. But you can also see that the hormone is having effects on empathy in these children. So when they were 8 years old, they were given that same test of empathy, but a child version of it, looking at photos of the eyes. And what we see is a negative correlation. The higher the child's prenatal testosterone, the more difficulties 8 years later they're having in reading faces to interpret what someone might be thinking or feeling. Here, the correct answer is that the person in the photo is interested in something. And we can see again in this normative sample that the same hormone seems to be correlated with performance on attention to detail. So now we've got a child version of that test. Can you find the triangle? I'll trace it out for you, hidden in the overall design. And the higher your prenatal testosterone, the faster and more accurate you are at seeing the part within the whole at 8 years old. But let me just go back one slide. You can see these are relatively small samples. These are women who've had amniocentesis, but it's unlikely that there's a child who's got a diagnosis in that sample because the samples are way too small. We heard earlier that autism is about 1% of the population. So in order to test whether prenatal sex steroids were associated with diagnosed or clinical autism, we collaborated with the Danish Biobank in Copenhagen because they've been collecting amniotic fluid from every woman in Denmark who has amniocentesis, storing it in their freezer, so it's a biobank. And on that basis, they've got almost 20,000 samples from pregnancies where the baby was born in the 1990s. And they also have a register, a psychiatric register for who gets a diagnosis of anything psychiatric. What we knew was that there were at least 128 of these babies had gone on to receive a diagnosis of autism. And of course, we could pick as many controls as we liked. So we picked about twice as many controls. And we looked at those same delta-4 sex steroids, which goes from progesterone through to testosterone. You saw that earlier. And what we found was that those who went on to receive a diagnosis of autism did have slightly elevated levels of exposure to prenatal testosterone compared to typical males, who in turn have higher levels than typical females. So this might be one of the non-genetic or epigenetic factors that are acting on the genetic predisposition to autism, and which may be relevant to explain why autism may be more common in boys. More recently, we looked at a different prenatal sex steroid hormone, estradiol, because animal research again suggests that this hormone has masculinizing effects on brain development in rodents, for example. And again, what we see is that in autism, there are higher levels of prenatal estradiol compared to controls. So you can see that we were looking at a whole bunch of estrogens, estradiol, estrone, progesterone, and 17-OHP. All of these showed elevated levels. You can see the odds ratio in autism compared to controls. How these sex steroids are thought to change brain development is seen in this kind of schematic cartoon where testosterone can cross the cell wall. It's aromatazed into estrogen. So under the influence of the enzyme aromatase, it's converted into estrogen, where it can bind to estrogen receptors, enter the nucleus, and function as a transcription factor to change gene expression, as we heard earlier. And the model posits that it then it reduces the rate of apoptosis. What's the evidence for this? Well, if you look at some of the classic studies done in mice and rats, if you take a female mouse and look at this part of the brain, it's called the sexually dimorphic nucleus of the preoptic area. This is what a female, typical female, that region of the brain looks like. And this is what a typical male looks like in terms. So you can see males have a lot more neurons and a lot more connections between neurons. But what's being shown in this slide is that if you take a typical female and give her extra estradiol, her brain looks more like the typical male than the typical female. So the hormone is having so-called masculinizing effect on brain development. Those are kind of classic animal studies. Here in Cambridge, there's a group at the MRC LMB, Laboratory of Molecular Biology, led by Madeline Lancaster, that is doing similar work, but in cerebral organoids. So these are stem cells that have been programmed to develop into neurons and are being grown as a 3D structure. I'm sure many of you have heard about this new technology. And what's being shown here is a control mini brain, as they're called, and a mini brain which has been given DHT, which is a potent androgen. And what you see is more of these neural precursors, excitatory neural precursors in the organoids that are exposed to higher levels of DHT. So this is a human model system showing sort of similar results to what we see in the animal studies. So just before we kind of come to a close, we might wonder, in the case of autism, where is the elevated testosterone or sex steroid hormones coming from? Is it the fetus, or is it the mother, or is it from a third source, which could be the placenta, because the placenta itself synthesizes sex steroids? What you can see in this Chinese study, published now 5 years ago, was that they took serum samples from pregnant women. They took serum samples from mothers of a child with autism and then mothers of typical children, but it was taken during pregnancy. And then, they followed them up to find out the diagnosis. And you can see that the mothers of autistic kids had higher levels of testosterone relative to mothers of typical kids. So the source could be maternal. And some more evidence that mothers may be contributing to this prenatal sex steroid effect comes from women who have polycystic ovary syndrome, a medical condition that's caused by elevated testosterone, and where the phenotype is that the woman has late puberty, so late onset of menstruation, irregular menstrual cycles, and excess bodily hair or hirsutism. And what you can see here is that women who have this syndrome and who therefore have higher levels of testosterone have an elevated likelihood of having a child with autism. And this has been demonstrated in 4 different studies in different countries. So it's the odds ratio of having a child with autism relative to a woman who does not have polycystic ovary syndrome, which would be an odds ratio of 1. So in fact, this is obviously a subgroup, a sort of special subgroup in the population, which can be used as a sort of experiment of nature to see whether the mother's levels of testosterone increase the likelihood of autism in her unborn child. I think this is my final slide, but again, back to animal models. Some recent studies showing that if you this is in rats, that if you expose a mother to the mother rat to higher levels of testosterone by giving her a drug, letrozole, which actually blocks the conversion from testosterone into estrogen. So you get a buildup of testosterone. You can see that that her offspring spend less time interacting with a conspecific, another rat, compared to a rat that's not given that drug, so just given effectively the placebo. So again, some evidence of changes in the offspring's behavior, a reduction in social behavior following administration of extra testosterone. So putting all this together, this is the sort of model that we're working with, that clearly autism has a genetic component to it. But we also know that during pregnancy, hormones are being released by the mother and by the fetus. You can see in the case of the fetus, shown in blue is the male fetus, where there's a surge in the production of testosterone. And shown in red here is the testosterone curve in female fetuses. And in darker blue, there is estrogen or estradiol. But these hormones interact with genetic predisposition to change brain development, perhaps leading to increased neuronal density or reduction in pruning. And I've included in this model the placenta, which we want to study in more detail, because we don't yet know whether the placenta is also playing a role. But what we do know is that in typical pregnancies, the placenta produces high levels of estrogens. And there's some initial data for placental abnormalities in pregnancies that later result in autism. So here are my conclusions: that autism may be an extreme of the typical male profile of brain morphology, and also at the psychological level in empathy and what I call systemizing or pattern recognition. And what we've seen is that prenatal sex steroids are associated with specific aspects of typical sexual dimorphism in the brain and behavior, and that children who later receive a diagnosis of autism are exposed to elevated prenatal sex steroids. And so sex steroids may be just part of the puzzle in understanding the etiology of autism. And I'm closing by giving you a link to our website at the Autism Research Centre. That's at the bottom of this slide, and thanking our funders, which includes the Autism Research Trust, MRC, and Wellcome. So I'll stop there. Not sure how we're going for time.
Yeah, pretty well. We've got a few minutes, I think, for questions. Thank you so much, first of all, Professor Barenkeim. That was extremely interesting, through a tour de force of the evidence, right up to the most cutting edge work which you're involved in. There's a couple of questions that have come in. First of all, if you're happy to answer.
Sure.
First of all, possibly slightly controversially, or thinking sensitively about this, but looking forward, is there any scope for this work to be involved in an earlier, like a diagnosis of autism, possibly when the mother has is pregnant, and looking further than that, is there any scope for treatment, possibly in a very early stage.
Yeah, so I agree it's a controversial area for many reasons. So the reason we're doing this research is not towards prenatal screening. It's really just to understand causes of autism and the mechanisms that are involved. I'd say that the sort of current state of the research is it wouldn't lend itself to screening, even if you wanted to do that. You saw the results, and what we're seeing is that in autism, there's a slight sort of elevation, but nothing that you could use in terms of sensitivity or specificity in predicting autism. More controversial would be the idea of a treatment, which may, I don't know, what might cross people's minds is should we be offering some kind of testosterone blocker to reduce levels of these prenatal sex steroids. It's controversial because autism is a disability, but it's not just a disability. It's also just a difference, and it goes under what's commonly called neurodiversity. Just like gender is part of diversity, and gender differences in the brain, controversial in their own right. We wouldn't necessarily want to be treating or changing brain development in relation to diversity, because diversity is important in the population and is valued. But also some autistic people are not looking for a treatment, they're looking for support. But these days, because autism is also recognized to be genetic, part of their identity, again, treatment isn't necessarily the goal, any more so than, one wouldn't want to have treatment for any other aspect of one's identity.
Yeah, that's a very nuanced answer. Thank you. A couple more questions. Have the recent increases in the diagnosis and recognition that females with autism present differently to males, has that changed the theory and the methodology used?
So I think it's probably too early to know. So what I showed in some of the early slides was that more females are being diagnosed, and that's good. So many of them were traditionally missed or overlooked. The research has to kind of catch up. So some of the prenatal sex steroid research has only been done in males because there were too few females to analyze in terms of statistical power. So we do need sort of research which is more balanced in terms of gender to see whether, for example, the sex steroid hormones have different effects in males and females.
Yeah, thank you. Someone's asking if you could please give an example of a proposed non-genetic cause of autism. And does the proportion of non-genetic and genetic causes vary in males and females with autism?
Yeah, I mean, separating genetic and non-genetic is not as easy as it sounds. So, for example, when I was talking about polycystic ovary syndrome, it's a hormonal condition in mothers, but in all likelihood, it's got a genetic basis. So it may be having non-genetic effects, so direct hormone effects on the fetus's brain, but it's ultimately arising from genes that the mother is carrying. Another example, maybe a sort of clearer non-genetic example, is gestational diabetes. So women who are pregnant and who are gaining weight too quickly and who get diagnosed with gestational diabetes have a higher likelihood of having an autistic child. Again, that's probably relevant to this sex steroid theory because gestational diabetes involves dysregulation of insulin, but the knock-on effect of dysregulation of insulin is also to dysregulate testosterone. They seem to kind of travel together. So, these might be seen as examples of the uterine environment, if you like, having an effect on autism likelihood.
Great, and kind of similar to that, but maybe moving away from autism, is there any link between the sex steroid theory and other conditions such as ADHD perhaps, or Tourette syndrome?
Right. No, it's a really good question. So in our studies, we've only looked at autism so far. We are seeking funding to see whether this is specific to autism or if it would be true, if it would be seen in other conditions that are also more common in males. So you mentioned ADHD, which again is more common in boys, and Tourette's. And my guess is it won't end up being specific to autism, but we have to wait for the data.
Great. Yeah, I think that's a nice place to end, given the time as well, looking ahead. Thank you very much, Dr. Barrington. And there's a message in there in the chat from Leo Penrose to you, who was actually involved in one of your studies. So that's it's really great that more people are being educated about this, and it's such an important topic.